we are going to talk about amino acid, its structure and function, and its acid-base properties. Amino acid, any of a group of organic molecules that consist of a basic amino group, an acidic carboxyl group, and an organic R group, or what we call the side chain, that is unique to each amino acid, is the term which is short for alpha amino carboxylic acid. Each molecule contains a central carbon atom, or what we call the alpha carbon, to which both an amino and carboxyl group are attached. The remaining two bonds of the alpha carbon atom are generally satisfied by a hydrogen atom and the R group. This is the formula of a general amino acid. The amino acids differ from each other in the particular chemical structure of its side chain, or the R group. When we hear the word proteins, we immediately remember these foods. Amino acids are building blocks of these proteins. Proteins are of primary importance to the continuing functioning of life on Earth. Proteins catalyze the vast majority of chemical reactions that occur in the cell. They provide many of the structural elements of the cell, as they also help bind cells together into tissues. Some proteins act as contractile elements to make movement possible. Others are responsible for the transport of vital materials from the outside of the cell, or the extracellular part, to its inside part, which is the intracellular. Proteins in the form of antibodies protect animals from disease, and in the form of interferon mount an intracellular attack against viruses that have eluded destruction by the antibodies and other immune system defenses. Many hormones are proteins as well. Lastly, many proteins control the activity of genes, which what we call the gene expression. The simplest amino acid is called the glycine, named for its sweet taste, which is a term for sugar, what we call the glyco. It is one of the first amino acids to be identified, having been isolated from the protein gelatin in 1820. In the mid-1950s, scientists involved in elucidating the relationship between proteins and genes and agreed that 20 amino acids, or the standard amino acids, were to be considered the essential building blocks of all proteins. The last to be discovered is the threonine. It has been identified in 1935. Let's talk about chirality. All the amino acids but glycine are chiral molecules. That is, they exist in two optically active asymmetric forms called enantiomers that are the mirror images of each other. This property is conceptually similar to the spatial relationship of the left hand to the right hand. One enantiomer is designated D and the other L. It is important to note that the amino acids found in proteins almost always possess only the L configuration. So this reflects the fact that the enzymes responsible for protein synthesis have evolved to utilize only the L enantiomers. Reflecting this near universality, the prefix L is usually omitted. Kaya do understood naman nga L lang ang ga-occur, or almost always. And some D amino acids are found in microorganisms, and they are particularly in the cell walls of bacteria and in several of the antibiotics. And let's jump to the acid-base properties. Another important feature of free amino acids is the existence of both a basic and an acidic group at the alpha carbon. Compounds such as amino acids that can act as either an acid or a base, they are called amphoteric. Next, the pKa value is one method used to indicate the strength of an acid. pKa is the negative log of the acid dissociation constant, or the Ka value. A lower pKa value indicates a stronger acid. That is, the lower value indicates the acid more fully dissociates in water. The pH naman is a measure of the concentration of hydrogen ions in an aqueous solution. Essentially, pKa tells you what the pH needs to be in order for a chemical species to donate or accept a proton. The relationship between pH and pKa is described by the Henderson-Hasselbalch equation. The basic amino group typically has a pKa between 9 and 10, while the acidic alpha carboxyl group has a pKa that is usually close to 2. That is already a very low value for carboxyls. The pKa of a group is the pH value at which the concentration of the protonated group equals that of the unprotonated group. Thus, at physiological pH, about mga 7 to 7.4, the free amino acids exist largely as dipolar ions, or nag-carry as an individual or as an equal number of positive and negatively charged group, dipolar. Any free amino acid and likewise any protein will, at some specific pH, exist in a form of dipolar ions. That is, all amino acids and all proteins, when subjected to changes in their acidity, pass through a state at which there is an equal number of positive and negative charges on the molecule. The pH at which this occurs is known as the isoelectric point, or the isoelectric pH, and is denoted as Pl. When dissolved in water, all amino acids and all proteins are present predominantly in their isoelectric form. Or in other words, there is a pH, or there is an isoelectric point at which the molecule has a net zero charge, meaning there is an equal number of positive and negative charges. But there is no pH at which the molecule has an absolute zero charge, or ang complete absence of positive and negative charges. That is, amino acids and proteins are always in the form of ions. They always carry charged groups. This, in fact, is vitally important in considering further the biochemistry of amino acids and proteins. These are the essential and non-essential amino acids. The difference between essential and non-essential amino acids is that non-essential amino acids can be made by the body, while essential amino acids cannot be made by the body, so you must get them from your diet. You must have all of the amino acids so your body can build the wide variety of proteins it needs. and the standard amino acids. One of the most useful manners by which to classify the standard or common amino acids is based on the polarity, that is the distribution of electric charge of the R group or ang ginatawag na side chain. The group 1 or the non-polar amino acids, these are the glycine, alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, and tryptophan. The R groups of these amino acids have either aliphatic or aromatic groups. This makes them hydrophobic or water-fearing. In aqueous solutions, globular proteins gani will fold into a three-dimensional shape to bury these hydrophobic side chains in the protein interior. Isoleucine is an isomer of leucine and it contains two chiral carbon atoms. Proline is unique among the standard amino acids in that it does not have both a free alpha amino and free alpha carboxyl groups. Instead, its side chain forms a cyclic structure as the nitrogen atom of proline is linked to two carbon atoms. Strictly speaking, this means that proline is not an amino acid but rather an alpha amino acid. Phenylalanine, as the name implies, consists of a phenyl group attached to an alanine group. Methionine is one of the two amino acids that possess a sulfur atom. And methionine plays a central role in protein biosynthesis or the translation, as it is almost always the initiating amino acid. Methionine also provides methyl groups for metabolism. Next is tryptophan, contains an indole ring attached to the alanyl side chain. Group 2 or the polar uncharged amino acids are the serine, cysteine, threonine, tyrosine, asparagine, and glutamine. The side chains in this group possess a spectrum of functional groups. However, most have at least one atom. It's either nitrogen, oxygen, or sulfur, with electron pairs available for hydrogen bonding to water or other molecules. Two amino acids, serine and threonine, contain aliphatic hydroxyl groups, that is, an oxygen atom bonded to a hydrogen atom represented as an alcohol. Tyrosine possesses a hydroxyl group in the aromatic ring, making it a phenol derivative. The hydroxyl groups in these three amino acids are subject to an important type of post-translational modification. The phosphorylation, like methionine, cysteine contains a sulfur atom, and unlike methionine sulfur atom, however, cysteine's sulfur atom is very chemically reactive as it undergoes sometimes cysteine oxidation. So, asparagine, first isolated from asparagus, and glutamine both contain amide R groups, and the carbonyl group can function as a hydrogen bond acceptor. And the amino group naman can function as a hydrogen bond donor. Next, group 3, the acidic amino acids. The two amino acids in this group are aspartic acid and glutamic acid. Each has a carboxylic acid on its side chain that gives it an acidic property. In an aqueous solution at physiological pH, the pH 7 or up to 7.4, all three functional groups on these amino acids will ionize, thus giving an overall charge of -1. In the ionic forms, the amino acids are called aspartate and glutamate. The side chains of aspartate and glutamate can form ionic bonds, and they can also function as hydrogen bond acceptors. Many proteins that bind metal ions for structural or functional purposes possess metal binding sites containing aspartate or glutamate side chains or both. Free glutamate and glutamine play a central role in amino acid metabolism. And glutamate is the most abundant excitatory neurotransmitter in the central nervous system. Group 4 is a basic amino acid. The three amino acids in this group are arginine, histidine, and lysine. Each side chain is basic, or in other words, they can accept a proton. Lysine and arginine both exist with an overall charge of +1 at physiological pH. And here, the guanidino group in aspartate or in arginine side chain is the most basic of all R groups because it has a pKa value of 12.5. The imidazole side chain of histidine allows it to function in both acid and base catalysts or at near physiological pH values. None of the other standard amino acids possesses this important chemical property, and therefore histidine is an amino acid that most often makes up the active sites of protein enzymes. The majority of amino acids in group 2, 3, and 4 are hydrophilic, which means they are water-loving, and as a result, they are often found clustered on the surface of globular proteins in aqueous solution. Amino acid reactions. Amino acids, via their various chemical functionalities, can undergo numerous chemical reactions. However, two reactions, which are the peptide and the peptide bond and the cysteine oxidation, are of particular importance because of their effect on protein structure. In peptide bond, amino acids can be linked by a condensation reaction in which a hydroxyl group is lost from the carboxyl group of one amino acid along with a hydrogen from the amino group of the second amino acid, forming a molecule of water and leaving the two amino acids linked via the amide or via an amide called a peptide bond. At the turn of the 20th century, German chemist Emil Fischer first proposed this linking together of amino acids. Note that when an individual amino acids are combined to form proteins, their carboxyl and amino groups are no longer able to act as acids or bases, since they have already reacted to form a peptide bond. Therefore, the acid-base properties of proteins are dependent upon the overall ionization characteristics of the individual side chains or the R groups of the component amino acids. Amino acids joined by a series of peptide bonds are said to constitute a peptide. After they are incorporated into a peptide, the individual amino acids are referred to as amino acid residues. Small polymers of amino acids, which are fewer than 50, are called oligopeptides, and those naman that are larger than 50 or more than 50 are referred to as polypeptides. Hence, a protein molecule is a polypeptide chain composed of many amino acid residues, with each residue joined to the next by a peptide bond. The lengths for different proteins range from a few dozen to thousands of amino acids, and each protein contains different relative proportions of the 12 standard amino acids. Cysteine oxidation. The thiol or sulfur-containing group of the cysteine is highly reactive, as we've said earlier, and the most common reaction of this group is a reversible oxidation that forms a disulfide. Oxidation of two molecules of cysteine forms cysteine, a molecule that contains a disulfide bond. When two cysteine residues in a protein form such a bond, it is referred to as a disulfide bridge. Disulfide bridges are common mechanisms used in nature to stabilize many proteins. Such disulfide bridges are often found among extracellular proteins that are secreted from cells. In eukaryotic organisms, formation of disulfide bridges occurs within the organelle called the endoplasmic reticulum. In extracellular fluids such as blood, the sulfhydryl groups of cysteine are rapidly oxidized to form cysteine. In a genetic disorder known as cysteinuria, there is a defect that results in excessive excretion of cysteine into the urine. Because cysteine is the least soluble of the amino acids, crystallization of the excreted cysteine results in the formation of a calculi, more commonly known as stones, in the kidney, ureter, or urinary bladder. The stones may cause intense pain, infection, and blood in the urine. Medical intervention often involves the administration of dipenicillamine, it works by forming a complex with the cysteine, and this complex is 50 times more water soluble than cysteine alone. In summary, it is the sequence of amino acids that determines the shape and biological function of a protein, as well as its physical and chemical properties. Thus, the functional diversity of proteins arises because proteins are polymers of 20 different kinds of amino acids. For example, a simple protein is the hormone insulin, which has 51 amino acids. With 20 different amino acids to choose from at each of these 51 positions, a total of 2,051 or about 1,066 different proteins could theoretically be made. Other functions of amino acids are precursors of a variety of complex nitrogen-containing molecules. Prominent among these are the nitrogenous base components of nucleotides and the nucleic acids found in DNA and RNA. Furthermore, there are complex amino acid-derived cofactors such as Hem and chlorophyll. Hem is the iron-containing organic group required for the biological activity of vitally important proteins such as the oxygen-carrying hemoglobin, and the electron-transporting cytochrome C, chlorophyll, is a pigment required for photosynthesis. Several alpha amino acids or their derivatives act as chemical messengers. For example, gamma aminobutyric acid or the GABA, a derivative of glutamic acid, and serotonin and melatonin, derivatives of tryptophan, and histamine, which is synthesized from histidine, are neurotransmitters. Thyroxine, which is a tyrosine derivative produced in the thyroid glands of animals, and indole acetic acid, a tryptophan derivative found in plants, are two examples of hormones. Several standard and non-standard amino acids are often vital metabolic intermediates. Important examples of this are the amino acids arginine, citrulline, and ornithine, which are all components of the urea cycle. The synthesis of urea is the principal mechanism for the removal of nitrogenous waste. There are non-standard amino acids. Non-standard amino acids refer to those amino acids that have been chemically modified after they have been incorporated into a protein. This is called a post-translational modification. And those amino acids that occur in living organisms but are not found in proteins, these are also called non-standard amino acids. Among these modified amino acids is the gamma-carboxyglutamic acid, a calcium-binding amino acid residue found in the blood clotting protein prothrombin, as well as in other proteins that can bind calcium as part of their biological function. The most abundant protein by mass in vertebrates is collagen. Significant proportions of the amino acids in collagen are modified forms of proline and glycine, or the 4-hydroxyproline and 5-hydroxylysine. Genes. Genes are made up of promoter regions and altering regions of introns, or the non-coding sequences, and the exons, which are the coding sequences. The production of a functional protein involves the transcription of a gene from DNA to RNA. The removal of introns and splicing together of exons, the translation of the spliced RNA sequences into a chain of amino acids, and the post-translational modification of the protein molecule. Arguably, the most important post-translational modification of amino acids in eukaryotic organisms is the reversible addition of a phosphate molecule to the hydroxyl portion of the R groups of serine, threonine, and tyrosine. This event is known as the phosphorylation and is used to regulate the activity of proteins in their minute-to-minute functioning in the cell. Serine is the most common phosphorylated residue in proteins, and threonine is second, and tyrosine is the third. Proteins with carbohydrates or sugars covalently attached to them are called glycoproteins. Glycoproteins are widely distributed in nature and provide the spectrum of functions already discussed for unmodified proteins. The sugar groups in glycoproteins are attached to amino acids through either oxygen or nitrogen atoms. These are called respectively O-linked sugars or the N-linked sugars in the amino acid residue. The O-linked sugars are attached to proteins through the oxygen atoms in serine, threonine, hydroxylysine, or hydroxylproline residues. The N-linked sugars naman are attached to proteins through the nitrogen atom in asparagine. Finally, there is a case of selenocysteine, although it is part of only a few known proteins. There is a sound scientific reason to consider this the 21st amino acid because it is in fact introduced during protein biosynthesis rather than created by a post-translational modification. Selenocysteine is actually derived from the amino acid serine in a very complicated fashion, and it contains selenium instead of sulfur from the cysteine. The modern biochemist has a wide array of methods available for the separation and analysis of amino acids and proteins. These methods exploit the chemical differences of amino acids and, in particular, their ionization and solubility behavior. A typical determination of the amino acid composition of proteins involves three basic steps, which is hydrolysis of the protein to its constituent amino acid. Next is separation of the amino acids in the mixture. And lastly, quantification of the individual amino acids. Hydrolysis is accomplished by treatment of a purified protein with a concentrated acid solution at a very high temperature, usually around 110 degrees Celsius, for up to 70 hours. These conditions cleave the peptide bond between each and every amino acid residue. The hydrolyzed protein sample is then separated into its constituent amino acids. Methods important for amino acid separations include ion exchange chromatography, gas chromatography, high-performance liquid chromatography, and most recently, mayroon sang capillary zone electrophoresis. Some common uses of amino acids. The industrial production of amino acids is an important worldwide business. The first report of the commercial production of an amino acid was in 1908, and it was then that the flavoring agent monosodium glutamate, or the MSG, much more commonly known to us Filipinos as the bitsin, was prepared from a type of large seaweed. This led to the commercial production of MSG, which is now produced using a bacterial fermentation process na, with starch and molasses as carbon source. Glycine, cysteine, and D and L-alanine are also used as food additives. And mixtures of amino acids serves as flavor enhancers in the food industry. The amino acid balance of soy or corn protein for animal feed is significantly enhanced upon the addition of the nutritionally limiting amino acids methionine and lysine. Amino acids are used therapeutically for nutritional and pharmaceutical purposes. For example, patients are often infused with amino acids to supply these nutrients before and after surgical procedures. Treatments with single amino acids are part of the medical approach to control certain disease states. Examples include L-dihydroxyphenylalanine, or the L-DOPA for short, for Parkinson's disease, and glutamine and histidine to treat peptic ulcers, and arginine, citrulline, and ornithine to treat liver diseases. Certain derivations of amino acids, especially of glutamate, are used as surfactants in mild soaps and shampoos. D-phenylglycine and dihydroxyphenylglycine are intermediates used for the chemical synthesis of beta-lactam antibiotics. These are synthetic versions of penicillin. And then aspartame naman is a sweetener prepared for the individual component amino acids are aspartic acid and phenylalanine. So if you're familiar of the sweet and low, the equal and the splenda nga mga sweeteners. This is a recommended video clip to be watched on YouTube, The Origins of Life by the Khan Academy. And lastly, for our references, please refer to this slide. Thank you so much for listening.